Now that we've introduced the concept of a dihybrid, you now face the challenge of telling apart a dihybrid cross from a monohybrid cross. In other words, when presented with a genetics problem, you're going to have to figure out if there are two loci involved or only one. Let's work through some pointers to help you decide between a monohybrid cross or a dihybrid cross. Oh, that should say red at that point. Here's a very typical genetics problem type of setup where we have some wild type organism. Now in this case, wild type we're going to define as red. This could be flower color or something along those lines. And we have two different mutant lines, pure lines now. We have one that's pure breeding that has an orange color and one that's pure breeding that has a white color. Now the question is, are these mutations that underlie these phenotypes, are they alleles of the same locus? I.e., are these monohybrids? Is this a monohybrid cross when we cross the two of them together? Or are they alleles of different loci, in which case this is a dihybrid cross? So let's work this through. Now, from these first crosses, we can see that whatever is causing the orangeness is recessive to its corresponding wild-type allele, and whatever is causing the whiteness is also recessive to its corresponding wild-type allele. And we see a 3-to-1 ratio for both of these crosses, as we would expect, which is indicative of a monohybrid cross. There's only one thing that's mutant in this, and whatever it is recessive to its corresponding wild type, and same thing here. Now, there's two possible outcomes. It could be that these are alleles of different loci, or on the other hand, they could be alleles of the same locus. I'm just going to use A's and B's to discuss these two different options. If they were alleles of different loci, I might say, okay, let's make the orange a. And the white line, if it's a recessive allele causing this condition that happens to be a different locus than this one, then we would have to choose a different base letter. Now here's the key point that many students find challenging. These different lines are only mutant in one thing, which means they have wild-type alleles for every other locus in the genome. What that would mean is that this orange line would be homozygous dominant for the wild-type allele of what is mutant here. It also means that this white line would be homozygous for the locus that is mutant in this particular line. If this was in fact what was going on, then in the F1 we would have a genotype that is a dihybrid. Okay, just like we've been talking about with Mendel's example of yellow seeds and purple flowers. Now, this particular phenotype now, in this instance, would be predicted to be wild type, which would mean red. Well, why is it wild type? Well, we have a big A, which means it's not going to be orange, and we have a big B, which means it's not going to be white. So this would have a wild type red phenotype. Now, this would then segregate just like we've been talking about. When we do an F1 times F1 cross, we would end up with that 9-to-3 to 3-to-1 ratio in the F2 that would take the following form. Now, we're going to talk about this later on if you're concerned about this genotype here. We've never seen both mutant phenotypes together, and how an organism could be simultaneously orange and white is something that we're going to deal with later. The point that I'm going to make now, however, is that in the F2, we will always see a wild type genotype, a wild type phenotype arising out of this particular F1. We will always see this wild type phenotype in the F2 if indeed we're dealing with a dihybrid in the F1. Let's now compare and contrast this with what we would expect if in fact these were alleles of the same locus. So just to be clear, this would have a red phenotype. We would expect redness to be there in the F2. There will be a wild type phenotype in the F2. Now I'll switch colors to help distinguish what's going on here. Let's now model this as a monohybrid cross and see what it would predict. So if this was a dihybrid, now let's choose a different symbol and model this as if it were a monohybrid. So same cross. Let's say we could stay with little a for the orange, except now if we're going to model this as a monohybrid cross, then this is mutant in the same locus that this one is mutant in. So we could call this some other allele. In this case, I'm going to use a lowercase a and put a superscript W to remind me that this is white, and I could put a superscript O here to remind me that this is orange. Now, a couple of things will happen here. In the F1, we would not have a dihybrid, we would have a monohybrid, and it would have one of each allele. And then in the F2, and here's the important difference, we would have a 1 to 2 to 1 ratio, just like you've learned for a monohybrid cross, except now we would never see wild type. There's no way to make a wild type genotype here. And in this case, what would the wild type be? Well, we could define the wild type as either a big A or perhaps a lowercase a with a plus. It's very common to represent a wild type using a plus in certain organisms, but a big A would suffice as well. But the point is that we'll never see red here. This would be orange, this would be white, this would be with whatever phenotype this happened to be. And we haven't specified what its phenotype would be at this point. But the point is that it wouldn't be red. It would either be orange or white, or if there's incomplete dominance going on, there'd be some sort of blending going on. So here's the key diagnostic. If it's a dihybrid cross, you will see wild type in the F2. If it's a monohybrid cross, you will not see wild type in the F2 because there's no way to generate a wild type genotype in the F2